Hello viewers, in this video today we are going to see about apoptosis. Now let's see introduction about apoptosis. Apoptosis is a natural programmed process where cells intentionally dismantle themselves to clear out damaged or unnecessary cells. During this process, the cell's genetic material fragments, the cell shrinks and it breaks apart into tiny vesicles known as apoptotic bodies. Every day, adults lose between 50 to 70 billion cells through apoptosis, while children aged between 8 to 14 experience the death of about 20 to 30 billion cells daily. Coming to the history of apoptosis, in 1842, Carl Vogt introduced the concept of apoptosis. 3 years later, Walter Fleming offered a detailed account of programmed cell death. In 2000, Kerr and Robert Horowitz were honored with a prestigious award for their groundbreaking research on apoptosis. Their research was conducted using the nematode worm Caenorhabditis elegans, a simple organism widely used in genetic studies due to its transparent body and well-mapped genome. Study this worm allowed the researchers to uncover fundamental biological processes that are conserved across species. Why do cells undergo apoptosis? Most cells are provided with an inbuilt mechanism of apoptosis as a part of the cell cycle. This mechanism allows the body to get rid of unnecessary cells or infected cells. Apoptosis is considered a vital part of various processes including normal cell cycle, proper development and functioning of the immune system, embryonic development and chemical-induced cell death. It is a part of development as it is essential in the differentiation of a mass of tissue into various groups. Apoptosis occurs in cells that might have been infected with viruses or might even be cancerous. This process usually takes place when the cell detects defects in the DNA and is not able to repair it. Apoptosis is also an essential part of the immune system as it clears the pathogen-specific immune cells once the foreign particle is removed from the body. This also helps to remove the immune cells that might react against the body's cells and cause autoimmune diseases. Another reason for apoptosis is to maintain homeostasis in the body by removing old cells to make space for the new ones. Now we'll move on to mechanism of apoptosis. Apoptosis is initiated by two primary stimuli that activate distinct but interconnected pathways. One is extrinsic pathway, the other one is intrinsic pathway. The extrinsic pathway is triggered by external signals. These signals typically come from other cells or the extracellular environment, such as binding of death ligands, example fast ligand or TNF-alpha, to specific death receptors on the cell surface. This interaction set off a cascade of events leading to apoptosis. The intrinsic pathway, on the other hand, originates from within the cell itself. It is usually activated in response to internal stress signals like DNA damage, oxidative stress, or mitochondrial dysfunction. This pathway involves the release of pro-apoptotic factors from the mitochondria, such as cytochrome C, which then promote the apoptotic process. Both pathways converge on the activation of a family of cystine proteases known as caspases. Caspases plays a crucial role in executing apoptosis by cleaving specific cellular substrates, leading to the orderly dismantling of the cell. Caspases are initially produced as inactive precursors called procaspases. Upon receiving apoptotic signals, procaspases undergo proteolytic cleavage to become active caspases, which then propagate the death signals through a cascade effect. Thus, this tightly regulated mechanism ensures that apoptosis occurs in a controlled manner, preventing damage to surrounding tissues and maintaining cellular homeostasis. Now first we'll see about extrinsic pathway. The extrinsic pathway of apoptosis initiated outside the cell, typically at the cell surface, where external signals triggers programmed cell death. This pathway begins with the activation of specific death receptors on the cell membrane, such as FAST receptor, tumor necrosis factor receptor, death receptor 3, and death receptor 4 and 5. These receptors are part of the tumor necrosis factor receptor superfamily and play a crucial role in sensing extracellular death signals. Activation occurs when death ligands such as FAST ligand, tumor necrosis factor alpha, APO3 ligand, and APO2 ligand bind to their corresponding death receptors. This ligand-receptor interaction is essential for transmitting the apoptotic signal into the cell. Upon ligand binding, the death receptor recruits adapted proteins and forms a multi-protein complex known as the death-inducing signal complex, and this serves as a platform for the activation of initiator caspases, particularly caspase 8. Once the death-inducing signaling complex is activated, it triggers the same effector caspase cascade as the intrinsic pathway, leading to the activation of caspases 3. These caspases arrange the dismantling of cellular components peaks in apoptosis. This convergence of extrinsic and intrinsic pathways ensure a robust and regulated mechanism for cell death, which is vital for maintaining tissue homeostasis and eliminating damaged or harmful cells. The another pathway is called intrinsic pathway. The intrinsic pathway of apoptosis is initiated from signals originating within the cell itself, often triggered by internal stressors. It is activated in response to various intracellular signals such as DNA damage, the loss of essential cell survival factors, oxidative stress, or other forms of cellular stress that threaten cell integrity. A critical regulatory mechanism of this pathway is the balance between pro-apoptotic and anti-apoptotic members of the BCL2 protein family. This balance determines whether the cell will proceed towards apoptosis or survival. Upon activation, pro-apoptotic proteins like BACs and BAC promote mitochondrial outer membrane permeabilization, leading to the release of cytochrome C into the cytosol. Cytochrome C, together with apoptotic protease activating factor 1 and ATP, forms a multi-protein complex known as apoptosome. The apoptosome then recruits and activates pro-caspases 9, converting it into its active form, caspase 9. Activated caspase 9 initiates a cascade by activating downstream effector caspases such as caspase 3, which execute the cell death program by cleaving various cellular substrates. This tightly regulated cascade ensures that apoptosis proceeds in a controlled manner, preventing unwanted cell death and maintaining tissue homeostasis. Conditions characterized by suppressed apoptosis can lead to various diseases including colorectal cancer, liver, lymphoidal malignancies, neuroblastoma, prostate, follicular lymphomas, carcinomas with p53 mutations, and autoimmune disorders such as myasthenia gravis, systemic lupus, erythematosus, and inflammatory diseases such as bronchial asthma, inflammatory bowel disease, pulmonary inflammation, and various viral infections such as herpes viruses, pox viruses, adenoviruses, baculovirus, and cowpox. Conditions characterized by excessive apoptosis can also lead to various diseases including AIDS, neurodegenerative disorders such as Alzheimer's disease, epilepsy, Parkinson's disease, retinitis pigmentosa, and cerebellar degeneration. Thank you viewers, thank you for watching the video.